Good evening, everybody. Good evening, and welcome to yet another supremely amazing lecture on the chapter Locomotion and Movement. This is your favorite channel, Biopoint. Stream the world of bio in front of you with yet another brand new topic, that is the structure of striated muscles. Give me hearts on the screen if everybody have joined. Give me hearts on the screen. I hope I'm audible to each one of you very clearly. The video is clear. Yes, everything is fine. Thank you so much. So this is the lecture number 2 on this amazing chapter of locomotion and movement. So this is a wonderful Telegram group at Biopoint New. It is a small family where we are discussing our doubts. Then regular OMR sheet examinations are done. Daily practice question papers, daily polls, quizzes, everything is done in our this small Telegram group. Don't forget to like, share, subscribe, and leave the comment in our wonderful Telegram group, as well as join our Telegram group. It's also okay. Those who haven't yet subscribed our wonderful channel, don't forget to like, share, and subscribe our channel for latest updates about and classes on Neet 2021-2022. Why no response on the chat box? Why no response on the chat box? What happened? Nila, Shivani, Sujana, Shraddha, what happened? No response on the chat box. Are you all here? Yes. Thank you so much. So today's our supremely amazing topic is the structure of striated muscles. So we are moving on with the structure of striated muscles. Okay. So back into the class, listen everybody. So skeletal muscle, what do you mean by striated muscles? What do you mean by striated muscles? In the yesterday's class we studied, right? Striated muscle means those skeletal muscles, right? What are striated muscles? Skeletal muscles. That is those muscles, those kinds of muscles which are voluntary and they are attached with the skeletal system or associated with the skeletal system of our body, right? So the skeletal muscle is made up of many muscle bundles, and that muscle bundles you will call it as fascicles. The muscle bundles you will call it as the fascicles. Clear? Look, this is a muscle bundle in which different muscles are constituted together, so that you will call it as the fascicle. The fascicles are held together by a collagenous connective tissue layer, which you will call it as the fascia. Look, they are held together. Look, a group of fascicles are held together by a structure, a connective tissue called fascia. Okay, so fascicles, fascia. Each fascicle consists of many muscle fibers. Each fascicle consists of many muscle fibers. Okay. So we studied about three different parts. That is, a skeletal muscle is made up of muscle bundles called fascicles. Then each fascicles are held together by a collagenous connective tissue called the fascia, and each fascicle contains many muscle fibers. Clear? I hope you all have liked the session. Those who haven't yet liked the session, don't forget to like it right now. So the muscle fibers are lined by plasma membrane. So generally you call it as the outer covering of the cell, you call it as the plasma membrane. But the covering, the plasma membrane of the muscle fibers, you call it as sarcolemma, which is enclosing exactly like the cytoplasm, you will call here as the sarcoplasm. Clear? Fascicle, fascia, sarcolemma, and sarcoplasm. Give me thumbs up on the chat box right now if you are clear with these 4 terms. First is the fascicle. Fascicle is a group of muscle bundles, and a group of muscle bundles are enclosed in a togetherly in a sheet called fascia. Then the plasma membrane, you will call it as the sarcolemma, and the cytoplasm in a cell, in a muscle cell, you will call it as the sarcoplasm. Thank you so much. Each muscle fibers contains parallelly arranged myofibrils or myofilaments. Very important. Okay, each muscle fiber consists of parallelly arranged myofibrils or myofilaments. That is an important property of the muscles. Clear? Sarcolemma, it is the plasma membrane. Then myofibrils or myofilaments are also present. Okay, clear everybody? If you take a structure of a muscle, then the outermost layer you will call it as the epimysium. The outermost layer you will call it as the epimysium. The middle layer you will call it as the perimysium, and the inner layer you will call it as the endomysium. So 3 layers you have to study: epimysium, perimysium, and endomysium. So these are the 3 different layers or 3 different parts of a skeletal muscle. Clear? Outer layer epimysium, perimysium, and endomysium. Clear? Then they consist of parallelly arranged cells, which you will call it as the myofilaments or the myofibrils. Clear? Next one. Yes. Each myofibril has alternate dark band and light band. Okay, so what are myofibrils? The parallelly arranged filaments is called myofibril. So each myofibril or myofilament has alternate dark and light striation. Okay, dark band and light band. So the dark striations present in the skeletal muscle you will otherwise known as anisotropic or A band. So a skeletal muscle mainly consists of two different kinds of striations are present in a skeletal muscle. First one is the dark band, and the second one is the light band. Dark band and the light band. Okay, guys, have you heard about the news that Neet is going to be conducted again day after tomorrow? I hope you might have read it in the newspaper, in the mobile news, etc. Neet is going to be again conducted for those students who haven't taken the test day after tomorrow once again, and the results is expected to be publishing on 16, that is this Friday. Okay, those who have written the test, they cannot write it once again. Okay, you have to go with the proof. So there are 2 kinds of filaments, that is a dark band and the light band. So guys, this is a previous year MCQ question. They will be twisting you and asking you the different terms. Dark band is also known as anisotropic band or A band. Light band is also called isotropic band or I band. Light, isotropic, light striations or isotropic, dark and anisotropic. Okay, so alternate light bands and dark band is constituting. So this is due to the presence of 2 fibrous contractile protein. Why a single striated muscles consist of more than 1 type of striations? This is a frequent question. Why it is so? Because of the presence of 2 different fibrous contractile protein. That is the actin filament and the myosin filament. So these are 2 different kinds of proteins, actin proteins and myosin protein. Clear? So we have to study whether dark band is made up of actin or myosin and light band is made up of actin or myosin, we have to study. Okay, which type of striations are constituting actin and which are myosin? That is our aim for this day. So once again, I am repeating the same thing. Myofibrils or myofilament has alternate dark and light striations. Anisotropic A band or dark band, light striations isotropic or I band, and this is due to the presence of two fibrous contractile proteins and thin actin filaments and the thick myosin filament. Guys, this is very important. Actin filament is thin and myosin filament is thick. First time I haven't seen those words that I was searching for. Okay, so the thin filament are the actin and thick filament is the myosin. Give me hearts on the chat box. Understood up to here? Give me hearts on the chat box. Thin actin, you have to remember like this: thin actin and thick myosin. Okay, clear. Thank you so much. So this is very important. Okay, so moving on. Okay. So the structure of striated muscles is the topic which we are discussing. So the I band, what are I bands? What are I bands on the chat box? Is it dark band or light band? Light band or dark band, which is called I band or isotropic band. On the chat box, guys, don't waste time. Fast, quick answers I need. What is the other? Yes, it is the light band. So light band is the I band, and I bands consist of actin. So I told you we have to study which type of band is constituting actin and which is constituting myosin. So I band contains actin, whereas A band, that is the dark band, contains both actin and myosin. Clear, everybody? Clear? Clear, everyone? So the light band consists of actin or the thin filaments and are called the I band, that is isotropic bands, and the dark band consists of both actin filament and myosin filament. Mostly they consist of myosin only. Okay, mostly dark band consists of myosin. Okay, according to your NCERT, it demands that. So if a question like that comes, you can write what? You can write what? Myosin. So I band consists of actin filaments and A band consists of myosin filaments, and the most important, they are arranged parallel to each other. They are arranged parallel to each other. Clear? Clear, everybody? So A band bears, that is the dark band, bears a lighter middle region. So A band or the dark band bears a lighter middle region, which you will call it as the H band. Which you will call it as the H band. Look guys, I'm going to explain with the help of this diagram. Guys, the blue colored portion is showing the I band and the dark colored portion is going to represent the myosin band or the A band. Clear? The green, look, the blue color represents, this all are the I band, and this brown color represents the dark band. Clear? So I'm going to explain it once again. A band, that is a dark band, in the center of the dark band, it bears a lighter middle region, which you will call it as the H band. Look, this is the H band, the lighter region. The lighter region, which you will call it as the H band or the H zone. H band, or you can call it as to the H zone. Clear? And guys, the definition for H band is it is a middle part of the A band that is not overlapped by the light bands. Okay, so the middle part of the myosin filament, that is the A band, which is not overlapped by the thin filament or the light band, is called H zone. Guys, if you look over here, it's clearly visible to you that this is the myosin. Oh my god. This is the dark band, and the dark band is definitely overlapped by the light band on both sides. Am I right? Look, the dark band is overlapped. The dark band is overlapped by a light band itself. But in the middle region, if you look in the H zone or the H band, you can see that the myosin filaments is only present. That is, it is formed only of myosin. So the actin filament will never enclosed or will never mount above this myosin filament. Clear? Guys, each and every point is very, very important. H zone, M line, A band, I band, Z line, all these are very, very important. So I have explained the third term, that is the I band. I explained isotropic band or the light band. A band I explained anisotropic band or the dark striations. H band, 2022 students also hearing this for the first time. Have you got out the point? Those who are hearing this for the first time, Shraddha, Shivani, Srujana, Nila, I think you have studied this earlier, right? So you can easily catch out of this. What about Shraddha, Shivani? Yes. So H band refers to the middle region of the thick band which is not overlapped by a thin filament. That is the H zone. So that is over. Next one, a thin dark line called the M line. A thin dark line called the M line runs through the center of the H zone. The myosin filaments lie in the A band and are held together in the middle by a thin filament, that is by a thin fibrous membrane called the M line. So this myosin and this myosin fragments are held together by this particular M line. Guys, is it clear? In your NCERT, the M line is like this. A single line is represented, but actually it's like this. But according to your exam, they will ask you this diagram. This is the enlargement of this diagram. We know that. So M line means the two myosin lines are held together by a dark line held together by a fibrous tissue that's known as the M line, which runs through the center of the H zone. Oh my god. Guys, here the weather condition is, just wait. Now is it okay for everybody? Please comment me on the chat box. Am I audible now? Here it is raining so heavily. Fine. If this issue happens again, please tell me. Okay. So that is the H band and the M line. Next one is, oh my god. The I band is bisected by a dense dark band called the Z line. Okay, so the I band is bisected by a dense dark band called the Z line. Okay, and the region between the two Z line, you will call it as a sarcomere. Clear? So I band is bisected by an Z line. Okay, I band is bisected. Look, this is the Z line. Okay, I band is bisected by an Z line, and the region between two is line that is starting from here to here, you will call it as the sarcomere. What are sarcomeres? Sarcomeres structure. I think again the problem is there. Am I? Sarcomeres are the structural and functional unit of muscle. Again, the one class is again lagging. So we have so guys, see you in the class sharp. Now is it audible? What about now? Now also the same problem. I hope now there is no issue. Once again, let's try. Okay. So sarcomeres are the structural and functional unit of a muscle. So generally we call nephron structural and functional unit of kidney, neuron structural and functional unit of nervous system. Exactly like this. Here we will call the sarcomeres. The sarcomeres are the structural and the functional unit of a muscle. Clear? Guys, when you observe the muscle tissue under the microscope, this is the structure that you are getting. So this is the dark line. This is the dark line. Look, M line. Clear? Structure. This is the Z line. This is another Z line. The structure between two Z line, you will call it as what? Sarcomere, which is a structural and functional unit. Yes. Then this zone, it's the H zone where the light band is not overlapping the dark band. Now you understood clearly what is meant by a dark band and what you mean by a light band. Look, this is light in color, that is light band. This is dark in color, so dark band. Clear? Next, we are going to discuss in detail about the structure of each protein, each contractile protein. First one is actin. What is actually actin? This is a diagram from your NCERT textbook. Very important diagram. So an actin is made up of two actin filaments. So actin is a polymer of two F actins, which forms a double helix structure. Okay. So actin is the protein. Actin is made up of two F actin, which forms a double helix structure. F actin is a polymer of the monomeric globular G actin. Clear? So actin is made up of two filamentous F actin. Actin is a polymer of two F actin, forming a double helix structure. F actin is a polymeric form of globular actin. Clear? So actin contains two other proteins called tropomyosin and troponin. Okay, Calvin? Tropomyosin and troponin. Neet 2022 students, those who are hearing this for the first time, take down all these things. Each statement is very important. Clear? 2 F-actin, and F-actin is the monomer of globular G-actin, globular actins or G-actin. And actin is mainly composed of two different kinds of proteins called tropomyosin and troponin. Okay? Clear? Next one. Oh my god. Yes. Two filaments of tropomyosin run along the grooves of the effactin. Okay. Two filaments of tropomyosin. Guys, this rope-shaped structure is called the tropomyosin. Look. This is one. I'll draw one more. This is another one. So two tropomyosin filaments are running along the grooves of the effactin. And the troponin is another protein, and troponin has three subunits which is located at regular interval. This is the troponin. This is the troponin which is found at regular intervals. So guys, look very carefully into this diagram. The green colored bead shaped portion, the bead colored portion, you will call it as the effactin. Okay, effactin. The rope shaped structure, you will call it as the tropomyosin, which is a protein. Then the bead like structure which are embedded like the jerry or the what? Cherry are kept on the top of a cake. Like that it is kept, it is called troponin. This diagram is a frequently at least 25 times repeated previous year question. Clear? Clear? So in the resting stage, a subunit in the resting stage, in the next class only we will be studying about the resting stage and all. Okay, when no muscle contraction is taking place, when all the muscles are in a relaxed manner, a subunit of troponin masks the binding site of myosin. So each myosin has certain binding site. So this troponin will bind, the troponin will mask the binding site on the actin filament, for myosin on the actin filament. So even though it is a myosin filament, it has certain site for attaching the actin filament. Clear? So here the troponin, which is a subunit of a troponin, will mask the binding site of myosin on the actin filament. So okay, sorry, I think I changed it. I interchanged it, right? Sorry. Clear? Once again I'll explain. In the resting stage, when all the muscles are in a relaxed manner, a subunit of troponin mask the binding site for myosin on the actin filament. So each actin filament has a site for attaching the myosin. Okay, so this troponin at the resting stage, this masks the binding site for myosin on the actin filament. Next is the myosin. So each myosin. So guys, I'll once again summarize all the things about actin once again. So each actin filament is made up of three type of proteins, namely F-actin, tropomyosin, and troponin. Okay, three different kinds of protein: F-actin, tropomyosin, and troponin. Each F-actin is a polymer of G-actin. What is the full form of G-actins? On the chat box, anyone? What is the full form of F-actin? G-actin. I'll read it. You can comment me. Okay. So two F-actins are helically held together. Okay. And through the F-actin, what happens? The tropomyosin, two filaments of tropomyosin, runs very close to the F-actins along their length. Clear? And the troponin, which is a complex protein which is present on the top intervals of the tropomyosin, and its subunit acts as a binding site for masking the active site of myosin on the actin filament. Absolute one is the globular actin. Globular actin. Am I audible? Am I audible? Am I audible? Yes. Next is the myosin. The myosin filament is a polymer of many monomeric protein called meromyosin. Okay, myosin is a polymer of monomeric proteins called meromyosin. A meromyosin has two main parts, that is the HMM or the heavy meromyosin, or you can call it as a cross arm, and a light meromyosin or LMM, which you can call it as the tail. What were the different parts of the actin filament? Effactin, then tropomyosin and troponin. And what are the different parts of the myosin? Heavy myosin, which also known as the cross arm, light meromyosin, which is otherwise known as the tail. Okay. So this is very important. What are the materials that constitute heavy meromyosin? It is a globular head plus the short arm. Light meromyosin constituted by the tail. This diagram is a frequently asked diagram in each exam. Again, cracking in the voice. Yes. A small cracking, right? Only one more slide left out in the today's class. Only one more slide, guys. One more. Okay. So this is a frequently asked question. Guys, this is the head region. This is the head region. This is the cross arm, which you will call it as the heavy meromyosin. Okay. And this is the tail. This is the tail region. And guys, one of the most important and a frequently asked question, guys, this top portion, look. Guys, very carefully you have to look. This two top portion, you can call it as the actin binding site. Actin binding site. Clear? And this two regions which I am drawing with a black pen, it is the ATP binding site. It is the ATP binding site. Clear? So this is the actin binding site and this is the ATP binding site. The heavy meromyosin consists of head region and the cross arm, and light meromyosin consists of the tail region. The globular head, that is a head region, consists of an active ATPase enzyme, which is used for, it has active binding sites for ATP and have active site for actin. That is, we have studied actin binding site and ATP binding site. So a particular enzyme is present which helps in the active binding of ATP, that is the ATPase enzyme, which is an important question for your NEET 2021. Guys, how was yesterday's biology exam? How was yesterday's biology exam? Was it so difficult? Was it so difficult? Tell me frankly, because I have to set the question for the next exam. If it is easy, the next exam should be the most difficult questions. If it was difficult, next question, easy questions from previous years. Tell me frankly on the chat box. Fourth question I have explained, right? Fourth question I think I have explained. Atherosclerosis and arteriosclerosis. Exactly what you also discussed in the group. It's the same. Actually, it was a thing. What? Interchangeable words. Atherosclerosis, arteriosclerosis. Clear? Yes, B and C. Those who have answered B or C, I'll give you the mark. Perfect. Yes. So guys, prepare for tomorrow. Read your NCRT. Diagram based questions will be there. Okay. And attend today's evening class. Most probably I'll try to have the class in the evening. Now itself it's the cracking in the voice, but 99% I'll try. 99% I'll try to schedule the class. Okay. So thank you guys. Thank you guys. See you in the next class. Oh my god. Before that, I have to show you something. Guys, this is something which we'll be taking in the evening session, body fluids in circulation, the question series. We'll be discussing the previous year questions. Clear? Bye-bye, guys. Take care. See you sharp at 8:30 today.